listen only mode. We're going to give folks a little bit of time to come in. It's not quite at the hour yet. So for those of you who are here early, I have a special treat for you. I bring you a microbe juggling a basketball. Not juggling, sorry, spinning a basketball. Isn't that outstanding? This is a ciliate that is using its cilia, its little motile hairs, to create a vortex and pull bacteria down its goozle. And this algae that is too big to swallow keeps getting sucked in there and blocking its aperture for eating, and it keeps kicking it out, and yet the vortex keeps bringing it back, and it looks just like spinning a basketball. Delightful. Brought to us by James Weiss, a great, I don't know if microscopist is a word, but if there are microscopists, he is a microscopist, if there ever was one. So good morning. Welcome to the presentation. My name's Scott McElveen. I am a technical sales rep here at Biome Makers, and I'm joined by my colleague, Sunny. Sunny, will you introduce yourself, please?
Yeah, happily. Good morning, good afternoon, maybe even good evening to some of our attendees. Thanks for joining our webinar today. My name is Sunny Kircher, based in Colorado, one of the key account managers at Biome Makers.
Thank you all for joining us. Today's topic is crop protection. Crop protection is a huge umbrella that ranges from everything. It includes weeds, it includes environmental stress, it includes all manner of insects, from the aphid group to sucking insects, mites, leafhoppers, sharpshooters, thrips, whiteflies, lace bugs, chewing insects like this corn earworm, beetles, caterpillars, snails, Colorado potato beetles, the kings of chewing insects, the grasshopper and locust groups. It includes microscopic enemies of plants. Root-feeding nematodes are known for their stylet with this knob at the end, which lets them generate the force to punch through the difficult to penetrate cell walls of plant roots. Here we have the classic poster child picture of a root knot nematode that has busted into a plant root and embedded its head inside. This is a tomato plant that has endured infection for 11 weeks from a infection of root knot nematodes. Potato cyst nematodes. Crop protection includes fungal pathogens, fungal diseases. Here's Phytophthora root and crown rot, which is common in many fruit and nut trees as a soil-borne fungus. Verticillium wilt would, of course, be the most famous pathogen. Potato pathogen, also a soil-borne fungus. Pythium, fusarium can cause devastating losses in corn and soy. Again, soil-borne fungal disease. White mold in soybean. All of this is under crop protection, including bacterial diseases. Here we have Pierce's disease in grapevine, and there are viruses, viroids, phytoplasmas. Our poor plants have a lot to deal with out there. But crop protection is a space that is seeing a great deal of pressure to innovate. It is experiencing new innovations. Sunny, what are the main principal drivers of driving innovation, pressures driving innovation in crop protection these days?
Yeah, well, clearly there's a lot to innovate around, right? These pathogens and pests are evolving always, just like us, just like plants. And here's a handful of different pressures that are impacting the innovation of crop protection products. So first is the diminishing efficacy due to resistance. So natural resistance over time, as these pests and pathogens evolve and kind of get to know the crop protection product and find their way around it one way or another. Slowed rate of discovery. So there's a lot of challenges around developing novel compounds and ultra low dose actives, degradable compounds, selective toxicants, and novel modes of action. Regulatory pressure. This is a big one, of course, coming from federal, state level, and local level regulatory bodies. And there's been an increasing level of scrutiny on chemistries and different crop protection products, especially in the last 10 years. And it's just making it a little more complicated of how to develop and bring to market an effective product that also is going to meet the regulatory demands.
Mm-hmm.
economic pressure. The cost of running trials and through the discovery process all the way to bringing the actual product to market. There's a high cost to this, of course, related to all three of the previous points, and then also the economic pressure within at the farm gate. Of course, as we all know, this is a really tough year for farmers in North America, and margins are pinched, and every decision really counts as far as like the ROI and an investment in a product application, even just putting it out once. And with a lot of crop protection products, it does require multiple sprays potentially throughout the season to get that good yield at the end. So economic pressure, always the consideration in everything we're doing.
That's the one I'm running into the most with the growers
Yeah.
I work with. What is essential for me to produce a crop, and what is gravy? Is there any way that I can spend money more efficiently when it comes to, like, for a lot of broadacre crops, fungicide is expensive enough that they just use treated seed and they won't have a subsequent application of fungicide later in the season.
Of course, yeah, and trying to estimate the cost of the risk before it's at your footsteps. It's it's a challenging task. So next is consumer pressure. Of course, farmers, agronomists, everyone that's making the decision about whether or not a product is applied or a practice even is applied on the field there's a lot of pressure there. And additionally, growers and agronomists are the portfolio in front of them of options of how to optimize their production is ever growing. So that's a pressure in its own of just staying up to date on different choices. And then lastly, advancements in applied microbiology and ag tech. On the one hand, this is actually driving innovation. And on the other hand, it can actually complicate how to bring the product to market and also decide whether or not it's going to get applied on a particular field.
Yeah, 5 of these drivers are push factors. They're pressures on the contemporary way of managing crop protection. The last one, at least, is a positive pull factor, development of peptides, RNA tools, biocontrol agents that are showing promise for improved crop protection. So, Sunny, where does biome makers stand in terms of crop protection? Where do we stand in terms of this topic? What is our role?
Yeah, so maybe even before answering that question directly, I'll just give a very high level overview of who we are as a company. And for anyone that wants to learn more, know that we have a very lengthy online library of different webinars and resources and published papers that you can reference. So check that out on our website if you have questions coming out of this webinar about who we are as a company. But high level snapshot, these little black pins that you see around the world are our labs, 2 of which are our own labs, both in Davis, California and Valladolid, Spain, and the rest are partner labs. So we are collecting biological soil samples from around the world. We've worked in over 200 different crop types, 55 different countries, and we are analyzing the soil microbiome. And below you see a list of some of our partners.
So I'm not going to belabor this because chances are this is not your first webinar. You've heard this spiel before. I'll be very brief. We extract DNA from soil. We snip out the microbial fingerprint, the marker gene we need to find out who it came from. That enables us to get a very broad and quite comprehensive look at the organisms, bacteria, archaea, and fungi living in a soil. And having created those identifications, we then refer to our world-leading database of functionality for those organisms so that we can deliver to the customer, here's your soil's capacity to, say, produce auxin, cytokinin, gibberellin, various plant growth hormones, to mobilize plant nutrients into an available form, immobilize them so they're no longer available, or cause them to gas off from the system, to cause disease, prevent disease, so on and so forth. All of our reporting, all of our services end up covering nutrient cycling, plant health, and soil quality. And I'm not going to go into detail into all of them because there are certain ones we're going to focus on today, given the topic of today's webinar. Suffice it to say, some folks do standalone soil tests, which produce an individual soil report for that sample, or we'll look at some today that are a pair of tests for comparing one field to another or one time point to another. There are free online tools in our online portal for comparing samples. And and looking into the microbiology of each sample. And then finally, we have Bee Crop Farm, which uses a combination of zoning, remote sensing, and DNA testing to map microbiological function. We will look at examples of all three over the course of this webinar. As usual, I've crammed too much in, but we won't be dwelling for the umpteenth time on this popcorn farm in Nebraska. I have examples from many countries and states, or not many countries, a couple countries, many states, many crops, many objectives. So the last kind of framing piece for this webinar is, A, we're not looking at weeds. We're not looking at stress in particular today. We're going to focus this webinar on pests and pathogens. Secondly, we provide a soil test. So we're going to major on soil-borne diseases, soil-borne threats. That's not to say we'll have nothing to say about, powdery mildew or citrus greening, which is vectored by a psyllid. However, that will be somewhat tangential. And in the third chapter, it's very difficult to make a webinar like this where we know we'll be speaking to farmers, agronomists, folks in retail, input manufacturers, both of traditional chemistries and biocontrol agents, talking to folks who are involved in many crops and regions. We can't be experts on every pathogen and every region. What we can do is we can illustrate ways that we, with our knowledge, which is, say if you're an agronomist in Iowa and you know all about Iowa corn, you know more than I will ever know about Iowa corn. But I can illustrate how this tool works so that you can then take it and combine it with your expertise and make your capacities that much sharper. So if we say anything that is not quite true to your crop or region, write Sunny a vicious email, and then take the chicken, spit out the bone. No, write me the vicious email. I want to learn.
I'll take it.
And then lastly, this whole presentation, keep in mind the disease triangle. This is the background. We should have it in the back of our minds, the whole presentation. We will measure through DNA the presence and abundance of the pathogen. That is only one ingredient to disease. To have disease, you must have the conjunction, the confluence. The stars must align between the disease-causing organism, an environment that enables it to grow, to proliferate, to switch into infection mode, and then a plant that is susceptible to becoming sick. We will return to the triangle a few different times over the course of this presentation. So I've structured things on the basis of kind of three frameworks of increasing complexity. This will make more sense as we go. But the first level would be direct engagement. If there's a disease out there, I, the grower, I have to deal with it. I have to suppress it. I have to protect my crop directly. It's my job. What DNA can add to this is at least we see before we spray. See and spray is usually applied to weeds. This would be, you can see the presence of the pathogen and the disease risk before symptoms appear, even before you plant, even when you're deciding your rotations, so that you detect it before you go to the expense of knockback or you make decisions to avoid altogether. This
and Scott,
approach
before we move forward into all these frameworks, I want the chance to answer the question you threw my way of how can biomakers support in disease management. The first, very simple, is we can measure the abundance of the pathogen in the field. Next, we're measuring the abundance and total impact of all the other functions, and that's what the bee crop metrics are. So whether that's natural bioagents to support in suppressing disease, or phytohormones and stress adaptation compounds that will support not only the soil microbiome, but also the plant in growing. So keep that in mind as we work through these frameworks, that we're not only measuring disease, but also all of the beneficial components of the soil microbiome that help protect the plant from disease, and combining that together.
And that'll especially become relevant with framework number 3. Framework 1, I'm just gonna ignore several dozen metrics, and I'm only gonna look at pathogens just for simplicity's sake and see if I only had that from bee crop, how would that inform decisions? And we'll start with, in this case, I'm taking on the persona of a watermelon agronomist in the North Florida, South Georgia region. My biggest issue that my growers face is fusarium rot. It can be devastating to a watermelon crop. You can have zero yield whatsoever if Fusarium oxysporum infects your plants. They will die before they've yielded you a single penny. And so all I've done here as an agronomist, and this is a client of mine, I've sent in 2 composite samples from 2 fields. Field 1 on the left, field 2 on the right. This tool, what it does is you can put in 2 samples or groups of samples, and it will simply highlight these are the biggest scores that are higher in one sample and the scores that are higher in the other sample. So on the left, we have the scores that are higher on field 1, and on the right, the scores that are higher on field 2. Sonny, what stands out to you on making a decision about, beg your pardon, I know that my grower has X number of acres of a watermelon plant that's grafted onto a resistant rootstock. I want to know where I put those resistant plants and where I put my unresistant varieties, my susceptible plants. Sonny, what do you see that would inform my decision of where to put my grafted resistant plants?
Yeah, well, playing the role of the grower who is just seeing their first bee crop results for the first time, and we've got 2 different areas of the field, or 2 different fields rather, measured here. On the left here, I see the results from field 2 in green, and I see 60 score for dampening off fungal root rot, fusarium root, and fusarium wilt. Those all seem pretty high. Is that right, Scott?
Yes, these are all saying that field 1 is 60 points higher out of 100 compared to field 2. And
so higher disease pressure in field 2.
In this case, it actually is a score of 60 compared to a score of 0 when it comes to fusarium wilt, root and stem rot, crown and root rot. So I should back up. Field 1 is here on top. I have just clicked on one of those scores to see, okay, it's a 0 in field 2, it's a 60 on field 1. Each of these, I have, with 2 samples, made a pretty clear decision. I'm putting my resistant plants on field 1. Field 2 is much lower risk. I have greater security putting my ungrafted plants there. 2 samples. All I looked at was a disease, didn't look at anything else, and I made an economically highly consequential decision.
Did you make that graph for me? Where did that come from?
This graph came from, way to embody the customer. This came from the free tool in our online portal that you can use to compare samples. It's called the metric box plot tool. Your account manager can help you find it, or it's built into the portal. Yeah, there's information about it in the help center. I use it almost daily myself.
Cool.
One nice thing on the PDF report, it will actually tell you how frequently a disease is detected in a given crop. So for watermelon crops, we detect damping off in 99% of all watermelon samples to some level. Fusarium we detect in 80% of watermelon soil samples to some level. So it's very difficult to completely be free of these pathogens. This field number 2 with 0 is unusual, therefore. It's in the 20% of, where was wilt? Here was wilt. The 20% of watermelon fields that do not have detectable fusarium wilt in the sample. So now I'm going to move over to the Mississippi Delta. I'm an agronomist of soybean growers. Here we're using a different tool. Each one of these rows is a bee crop sample, a soil sample for a soybean field. I've got 20 fields here, and then each column is a disease. And so taking on my agronomist hat, I'm going to run through what I know about these diseases briefly, because we have plenty more to see. But Drexlera blight, minor, occasional disease, rarely causes major yield loss. Let's log that away for later. Charcoal rot. Okay, this can be a much more serious disease. It penetrates roots in the spring. Penetrating roots, that makes a soil test, bee crop, a good test for watching it. It invades vascular tissue. And then, it invades vascular tissue from the roots in hot, dry conditions of the reproductive period. Fusarium root rot also invades young roots. Actually, the exudates from the young growing plant are what cause the spores to germinate and then infect the roots. It likes cool, wet, saturated conditions. It is worse under compaction. We'll circle back to that when we're making some decisions in a minute. Phomopsis, or name was recently updated to diaport. It's a pathogen that overwinters mostly on soybean residue, can also be soil-borne, but it will mostly be splashed onto the plant by rainfall or spores carried by the wind. It likes warm, wet weather during pod fill. That accelerates infection. So this one field that's high risk for it, if it's gray, it's undetected. If it is bright yellow, it's very low levels. If it's lighter yellow, it is somewhat low levels. So detected in the yellow, but not at a high risk level. Orange is a medium risk level. It's the light red and bright red. Yeah, light red and bright red that would be substantially high risk levels compared to other soybean samples. So this one field that's in red here, I'm going to prioritize timely harvest from that field compared to all the others, because this is a disease that's only going to start doing most of its damage to yield late in the season. Especially if it's warm and wet, I'm harvesting this field first.
That makes
We've
sense.
got target spot. It overwinters on residue, just like the last we looked at. Is splashed onto lower leaves by rain. Warm, humid conditions support the infection. You can choose resistant cultivars, rotate, scout high-risk areas. Stemphyllium, weak, secondary pathogen. It'll opportunistically infect wounds. Pythium, okay, this is a serious pathogen, much like Fusarium. It likes cool, saturated soils. We'll return to it in just a moment. Phytophthora, I've got one field where it was found, and it was found at high levels. It also likes saturated soils, like Pythium and Fusarium. You're noticing common thread there with saturation, poor drainage, poor aeration, but it likes them warm. We will return to that in a moment as well. And then finally, Neocosmospora, easy to confuse with red root rot. It's soil-borne. It infects young roots in the first weeks of life. It is most prevalent in the area that we're working here, the Mississippi River Delta. And it is especially associated with continuous soybean production and soybean cyst nematode. So now that we've surveyed the diseases, thinking like an agronomist, how can I help my grower make the best decisions? I'm seeing at least these 5 fields that have no substantial soil-borne disease risks. These are the fields where in a year like this, high input costs, low crop prices, we are the safest, the most secure in pulling back on some of the crop protection sprays, some of our fungicide sprays that we had been intending under a normal program. These fields have only secondary or minor pathogens. So I would again think these are in the safer bucket to pull back, but also I know precisely where to scout, knowing what to look for, what symptoms, and under what conditions those symptoms would appear. That is an enormous savings on time efficiency and allocating the most precious resource, the most limited resource of an agronomist, which is time, being able to know what to look for and where. These fields all have at least one major pathogen risk, particularly Fusarium and Pythium are the ones that are relevant to these. These are ones where, because both of those like cool, wet, saturated soils, the easiest thing I can do is just delay planting on these fields. I can wait until temperatures warm up, and I can see to it that I prioritize on these fields for soil improvement on improved drainage, be that through, bias for biology, having a acute tillage event followed by biology that can build structure, build aggregation, but mostly I can enhance this through aggregation, potentially through cover crops. Again, this varies region by region. I know cover crops aren't feasible everywhere on every system, but I need some way to improve drainage to reduce these risks in the future. And then the last cluster would be, these have multiple risk sources, multiple major diseases. I might consider rotating. I might consider, again, late planting and prioritizing these fields at the top of the stack for soil improvements. There's my Mississippi River Delta, using bee crop test, using the heat map tool. Like I told you, I've slammed this thing full of case studies. This is cotton in Arkansas. These are field reports from our bee crop farm platform, the mapping platform. One of my fields doesn't have high risk for any disease. It's maxing out at 47 for seedling disease complex, whereas the other is high risk for boll rot, seedling disease complex, charcoal root rot. This is a case where if I'm prioritizing resources, I prioritize them to the field on the right. And at the time this project was done, the disease risk maps were not high resolution, so I wouldn't necessarily know where the hotspots on a field are. But now, Sonny, what could you do scout-wise now that the disease maps are high resolution? If I've got a medium risk score, I'm not really sure if it's worth expending on crop protection.
Well, this allows for precision management, just to apply the product in one area of the field, and also to get a real quantify the value of the risk. Is it 20% of the field or 80%? If you don't have the capacity to do a variable application of a product, then you can at least decide, well, is this a minority risk or majority?
Mm-hmm. Yeah.
Question for you, Scott. I'm just curious, putting back on the customer hat, what does the 82 mean there for like the Negrospora rot, or the 42 for Pythium? Is that how many counts that you found of that species, or what does that number mean?
Every bee crop score ranges from 0 to 100. 100 is the highest abundance we've seen of this pathogen in a given crop. Okay. And then the 0 is undetected. So just above 0 would be the lowest abundance of this organism we've seen in a given crop. This would be soybean. So on that scale, from the most of this pathogen we've seen in a crop to the least, this section here in orange would be at a 42 on that spectrum of what's possible in soybean production based on tens of thousands of samples.
So when I see red, it's risk, and yellow is mellow.
Yes, that's right. So this is medium risk here. This is where it's best to scout. Take preventative measures, Pythium rot. Make sure we're using treated seed, especially in this area. But I'm scouting, I get to save energy by knowing exactly where to go, where I expect symptoms first. And like you said, if you do have the capacity to spot treat something with a drone, for instance, man, I can treat just this area, maybe a bit over here with a drone, and not subject my whole field to the expense, the diesel, and the negative effect on my beneficial organisms of spraying broad spectrum.
Mm-hmm. So this is like scouting, but from the soil, not from above.
Yes, you could put it that way. So it makes sense. That's framework number 1. All we looked at was the pathogen risk. We didn't bring any other elements into the conversation. Framework 2.
What do we got for framework 2 then?
Framework 2, I'm not making it exclusively my responsibility to deal with the disease. I'm going to recruit help. I'm going to recruit bodyguards, mercenaries, and let them help suppress the disease. The motto here would be, the enemy of my enemy is my friend. Let's talk about it. So I'm going to look, yes, at disease risk. I'm also going to look at natural biocontrol agents. What is a biocontrol agent, Sonny?
These are the beneficials in the soil microbiome. And so what we're measuring here at Biomakers is strictly bacterial and fungal species that are going to support the plant, either by directly killing or reducing the population of a disease or pathogen, or protect the plant in some other measure.
Well said. There's 4 groups we report on. The fungicide agents. We can't see we can't see azoxystrobin and imidacloprid. We see organisms. We see DNA. So we see critters that kill pathogenic critters. These are the ones that kill pathogenic fungi. So for instance, trichoderma is probably one of the most famous families of fungicide agents in soil. Here we have a kind of a dim yellow outline. That's the Pythium. That's the disease-causing organism. And inside of it, it has been invaded and parasitized. The parasite has been parasitized by trichoderma that's eating it from the inside out. There are bacteria like Bacillus subtilis that also can suppress fungal disease. Our antibiotics used for medicine, they were invented overwhelmingly, if not exclusively, by microorganisms, because they're the ones that have had to live together and fight each other for millennia. And so here's penicillium, the famous inventor of penicillin. We have microorganisms that will kill root-feeding insects and other insects. These grasshoppers have been digested from the inside out by a fungus called Bavaria bassiana. We have BT. You're all very familiar with BT, Bacillus thuringiensis, which ended up having some of its functionality programmed into BT corn, but it produces a toxin that breaks down the gut lining of insects, and then it would proliferate in their blood. And then there are microorganisms as well that kill plant pathogenic nematodes. Most of them specialize on plant pathogenic nematodes in particular. Here is a fungus that creates, it's called Arthroboitrus, it creates traps and lures the fungus into them, where it then clamps down, paralyzes it, and as fungus are wont to do, eat it from the inside out. You can notice a common theme there.
I was going to ask the results that you have in the middle there. This is from a single soil test, and are you saying that the microbiome of this soil has a really high capacity to defend my crop from bacterial pathogens and pests or pathogens?
Bacterial pathogens, it has a very high capacity to do so. Fungal pathogens, it has a middling capacity to defend the crop. Nematodes and insects, root-feeding insects, are not going to face many headwinds in trying to attack your crop. Your crop's somewhat on its own, defending against those.
So this is basically like, it's like the immune system of the soil.
It is sort of like the immune system of the soil.
All right.
I'm going to keep immunity as my device for framework number 3, and I'll leave this as, you can call it ecological immunity, but it's combatants, it's bodyguards, it's the Secret Service.
Okay, we'll go with that.
I'm going to take a brief excursus because probably some of you are like me and you liked watching a cheetah chase down a gazelle on National Geographic. I like to know how the organisms kill each other. And so here's a brief detour, a tour of some organisms and the ways that these biocontrol agents kill the pests and pathogens that we want to keep our crops safe from. One way, this is not a lethal way, it's just competition. It's just take up space, take up resources, take up water. Here you have 2 soil aggregates. One is lousy with Fusarium oxysporum. It is all these white strands, that's Fusarium oxysporum. Both have been inoculated with equal amounts of Fusarium, but it is only proliferated on the left. And that is because the aggregate on the left was sterilized before inoculation. The one on the right, it doesn't have any particular biocontrol, biofungicide in it. It's just a living soil. So the more your soil is alive, the more there's straight up competition. There's just headwinds to an organism attacking, which is why some crop protection companies are taking greater interest in how can we follow our product with a biological solution that will then prevent it from regrowing. In that, over here, this was free real estate. How do we make sure that after we knock back the pathogen, we make sure the house is inhabited so it's not free real estate again? Here's an example of, if you can look at this middle picture, you'll see kind of a film of green and black on this root surface. That's an organism called Methylorubrum extorquens. It is completely covering the surface of the root. That makes it difficult to get to the root to infect it. It serves as a shield. Secondly, they're consuming the exudates, the root releases. So we talked earlier about how Fusarium oxysporum spores in our soybean field were awoken. They germinated because they sensed the root exudates from the plant. Well, those are getting eaten up and monopolized, consumed. They're not going to get out as far into the soil and trigger germination of the Fusarium. And then likewise, you've got little glowing green spots within the root cells. Methylorubrum extorquens will get into the plant where it triggers a plant's immune response throughout its system. The plant starts producing defenses that it wouldn't otherwise. It's nice to have your friends get you healthy and boost your immune response rather than your enemies. Another way, some of them suppress the pathogens just by taking up space. Others can directly chemically attack and suppress the pathogen. So this bottom row here, you have 4 different species of Fusarium that have been put on a petri dish, and they're alone. They just get to grow. Again, it's free real estate. On the top, on either side of the Fusarium, they put a Bacillus amyloliquefaciens species. Around here, we just call it back Amy. So if you don't like amyloliquefaciens, just say back Amy. And you can see the Fusarium is not as free to grow. If you had this organism living on your root, as it grew, Fusarium would have a much more difficult time getting there. And it's not just a matter of it can't grow there. The strands, the hyphae of these fungi that are in proximity to, I believe I have this backwards, my mistake. So this bottom image here corresponds, the bottom right corresponds to the upper left. Upper left is healthy strands, nice, straight, healthy strands of Fusarium. And then in the bottom, these are the way the strands look in proximity to the bacillus. They're distorted, they're damaged, they are struggling because of the chemical warfare that's happening. The bacillus will produce compounds such as fungicin or iturin. In the A section here, these are healthy hyphae again. And then those that are subject to the fungicin produced by a Bacillus subtilis, they get twisted. Their internal structures are destroyed. Their organelles get digested. They shrink. They're distorted. Over here, these that were subject to iturin, it was even worse. Cell walls disappeared entirely. The membranes degenerated. Their innards shrank, and the hyphae start to fray and tear apart. So we're talking about biocontrol. This is the sort of thing that we're talking about. Pseudomonas fluorescens. It produces hydrogen cyanide, which essentially does the equivalent of microbially garroting its victims by preventing them from being able to transport electrons to produce energy. When we talk about oxidative damage, we hear a lot about reactive oxygen species. I like to see things. I like to be able to visualize what it would look like. So here's a quick video of a ciliate that does not like oxygen conditions. It's not adapted to them. And in preparing the microscope slide, it has been exposed to oxygen, and its membrane just bursts open and its innards all spill out. When our biocontrol agents subject their enemies, our plant's enemies, to oxidative stress, it is causing damages like this. It damages their DNA, damages their proteins, damages their organelles. And if it disrupts their membranes, they virtually just split open. Here's another ciliate. It's been exposed to oxygen. It just starts to unzip. It's kind of sad to watch, but also gratifying in other ways, thinking about your crop's pests and pathogens succumbing to a fate like this. Again, credit to James Weiss for the brilliant microscopy. And then I probably need to jump through a few of these, but like I said, fungi that will eat insects from the inside out. Here's Morbivaria bassiana. There's an actinobacterium that produces a insecticidal neurotoxin. It's a whole family of them, but the most famous is this Sacropolyspora spinosa. It poisons, intoxifies insects' brains. On the nematode front, here's a bacteria, Pasteurella penetrans, that attaches itself to the cuticle of root knot nematodes, and it feeds on them, it sterilizes them. Sterilizing them is a big deal because the females can lay 50 to 2,000 eggs. So that can absolutely put the brakes on an infestation if you can infect and sterilize the nematodes. These nematode eggs are being eaten from the inside out by Clonostachys roseae. Clonostachys also can get into a plant and trigger its immune responses to nematodes. It can also remotely paralyze and degrade nematodes by producing enzymes that break down their bodies and their proteins. Here again, there's a healthy nematode egg on the left and one on the right that has been taken over, eaten up by Purpucillum lalacinum. And then as a last treat on this tour, I mentioned the trapping fungi earlier. Here's an actual video of a fungus that has, or a nematode that has been trapped by this fungus, which is now paralyzing it and eating it from the inside out. Credit once again to James Weiss for the video. So where does that bring us? So the end of excursus, end of tour. I hope you all enjoyed learning about lethality on the microbial scale. This study is a throwback to back when biomakers was a technology exclusive to vineyards. It used to be called WineSeek. This is a vineyard in Spain where there was Blackfoot disease detected at comparable levels throughout the entire vineyard. And yet it was only the northern sections of the vineyard that experienced symptoms. There were no symptoms in the south. We did also sequencing of the plant material, and found that even inside the plants in the north, there was the most abundant fungus in the plant was Dactylonectria, was the Blackfoot disease, and not so in the south. The only difference that explained why there were no symptoms in the south was a substantially higher abundance of trichoderma, our beneficial biocontrol fungus, in the south. It was acting as a bodyguard and essentially took plant susceptibility out of the equation. And so there was no disease. Same amounts, same disease risks, but because of thanks to biocontrol agents, no disease in the southern areas. So referring to our chart, we can give you the high risk of the pathogen if the plant is not susceptible, thanks to its own immunity or thanks to suppression of the pathogen by beneficial organisms, you will not have disease. I'm going to illustrate this with a brief grid. This is our mental grid. On the left-hand side, we have, is our pathogen abundant or not? On the top are our biocontrol agents, our natural suppression. Are they abundant or are they not? If the natural biocontrol agents are abundant and the disease is not, best case scenario, few pathogens, lots of natural protection. That's a disease suppressive soil. Some of you will recall this from a webinar we did last year about diagnostics. If there are few of either groups, that's free real estate. That is that soil aggregate we saw that when the pathogen arrived, it didn't face headwinds. It was able to just proliferate, take over. So there's no risk right now, but we can't nip future risks in the bud if a pathogen blows in, is brought in on boots or machinery, if it's brought in on seed. If we have high biocontrol, high pathogen abundance, that soil is fighting. It's an embattled soil. Our crop is not alone in that fight. So it is not a worst case scenario. We need to monitor the situation and see who's got the upper hand in that fight. Worst case scenario, many pathogens, few biocontrollers, our crop is on its own. It has to fight for itself, fend for itself. Or back to framework one, I have to deal with it. I have to spend money on it. I have to spend time on it as the producer. So Sonny, here's an example for you to look at. We are in cotton production in Southern Virginia. We have I've overlaid the fungal disease suppressive organisms in the embeds, but you've got disease risk in the base map and then fungal biocontrol on the embedded map. What quadrant will we land in here?
What quadrant, as in the quintile?
Yes.
Well, it looks like well, the fungicide agents are low. So in the second quintile for our microbiome, specifically for the seedling disease complex, and we have high risk of the actual disease. So high disease and low natural suppressants. So I don't remember the number of the quadrants, but there we go. Vulnerable
number. Yeah, this soil actually proved vulnerable because we had a very wet spring in 2025. Mm-hmm. Now we're having a very dry spring, but last year it was wet. And in fact, this grower saw a great deal of seedling disease complex on his operation. Unfortunately, his sampling for this project took place a little bit late into the spring, too late to take some preventative measures to avoid susceptibility to seedling disease complex. It was too late, for instance, to employ a seed treatment of a biocontrol agent. And in this case, he did suffer a substantial degree of seedling disease complex. Here we're going to use again our metric box plot tool to compare two samples. Sonny, what stands out here as the biggest differences between, on the left, our poor performing, unhealthy, low yield field, and our, on the right, high performing, stronger crop?
Yeah, so on the left, we have the top three highest metrics, but in the delta between the two fields are all disease pressure. So really high disease in this green field on the left with the poor performing crop. And then on the right, looks like we have fungicide agents, zinc transport equilibrium, so zinc cycling, and then overall microbial functionality are the top three. So looks like things are in order over in the purple field.
Yeah, it's not always this stark, but in this case, we had a very strong informed hypothesis that the biggest difference between these two fields is how they're managing disease. Much higher pathogens on the left field, much higher capacity to suppress pathogens on the right field. So it wasn't like a out of this world, very high score for fungicide agents, but a lot better than the very low where the pathogens were running the show, running amok, so to speak. So we had a vulnerable soil in the poor crop, a disease suppressive soil in the strong crop.
Mm-hmm. And there are times in these fields, Scott, where we'll see high fungicide agents, but also high fungal pressure, meaning the fungicide agents are showing up to protect the crop. But in that case, when you just have fungicide agents, but not a lot of disease pressure, it's just naturally in good balance and ready for what comes at it, but there's not necessarily an existing risk.
And we'll see an example of that as well in just a sec.
Oh, great. Sorry, I'm getting ahead of myself.
This was corn in Arkansas, by the way.
Okay.
So here's an example that will give us 3 different parts of the quadrant. These are potato fields. Each of these arrows is a sample. We've got Fusarium oxysporum abundance in the left chart, and it is at an average of 6 to 7% of all fungal sequences on fields on blocks A and B, and a much lower percentage, around 1% of fungal DNA sequences on block C. Verticillium is, golly, it is up around, it's very small, but I believe that's 13 to 16% on blocks A and B of all fungal DNA that we recovered was from verticillium versus a much lower percentage on block C. Now, we're in framework 2. Let's look at the biocontrol agents. Block A and B both had high disease risk of Fusarium and verticillium. Block A, does it have fungal biocontrol agents? Yes, that lands it in an embattled soil. Here, the recommendation would be we can reduce the rate of fungicide, and we're going to follow it with biostimulus, be that seaweed extract, protein hydrolysate, organic acids, microbe food like an algae extract, something that will stimulate the beneficial biologicals in the soil so that the organisms we can see are there. We empower them to better defend the crop. We provide them food that maybe the crop can't supply because it is weakened in its own immune fight.
Mm-hmm.
Block B, we had high disease risk. We have very low levels of natural disease suppression. So we need the normal rate of knockback, and we should follow it with a biocontrol inoculum or potentially be investigating, well, there are native microbes on my operation. Why aren't they active and living here? Is there something I need to remediate about the soil structure, aeration, pH, salts, something that I need to remediate so that my microbes, my beneficials can work for me? And then block C, we had low disease risk. We've got great suppression, best case scenario, profit when you have good natural capital, let your microorganisms work for you. And then I'll dash through this quick. We have Fusarium dry rot real high in the northwest here. And just begin to see the fungal biocontrol agents. I want to say starting to respond, but to know if it's starting, which direction of change, you need to have multiple time points. This looks like a vulnerable soil. It might be moving into embattled soil.
Or the other direction. We don't, as you said, we don't know without the time points.
And so this would be probably cause to test again, see if you can take some measures to enhance your native suppressive capacities of your soil, and test again, are we improving matters? Reverse situation, also potatoes, also Fusarium dry rot. The bottom field, high risk, top field, no risk. Early blight, high risk, similar pattern on the bottom field, low risk at the top field. High risk of charcoal rot on the bottom field, low risk on the top field. And then look at that. Here we have disease suppressive. We've got high levels of fungal biocontrol agents in the north. And then in the south, that epicenter of all that pathogen risk is the lowest levels of biocontrol agents. This to me looks like there is, if I were looking at a response, I would expect higher levels of biocontrol agents where the disease was not. This looks like I'm asking myself the question, I need to investigate, why are the biocontrol agents that are active everywhere else not actively suppressing disease in this one area? What is compromising their activity? And as a matter of fact, insecticide agents are following the same pattern. So this is a signal to me. I need to investigate what's holding back my natural biological potential here. Sunny, speaking of which, speaking of insects, do we detect insects or nematodes?
No, we do not. Bacterial and fungal species with a few archaea on the side.
Right. So while we look at bacteria and fungi that can suppress or kill insects and nematodes, here's an olive operation down in Argentina. We're showing high numbers of nematicide agents, nematode suppressing microorganisms, high numbers of insect suppressing microorganisms. But you'll need to combine that with other assays, other observations as to, do I actually have an insect, root-feeding nematode problem to know whether this is a pest suppressive soil or an embattled soil, and also what's happening in these patches of lower potential. And I will pass by this one for now because I want to get in framework 3. This is the framework I'm most excited about when clients and customers are ready to think and operate at this level of a framework, which is, how do we maximize crop immunity so that the disease is prevented altogether, whether the pathogen is present or not, by the crop being not susceptible because it is thriving. The plant microbe partnership is humming, it is powerful, and the crop is no longer susceptible to a disease. The motto would be, an apple a day keeps the rhizoctonia away, eliminates susceptibility from the disease triangle altogether. Like I said, we need all 3 ingredients to have disease. So I'm going to borrow University of Nebraska Lincoln's crop watch Venn diagram image of the disease triangle to show when we pull the host away, it doesn't matter what the environment of the pathogen are. If the host is not susceptible, you do not get disease. And so, Sunny, what is the most important difference, the most salient difference between plants and animals when it comes to dealing with threats, dealing with disease and pests?
Well, the biggest difference that's consistent in both of those groups is that animals can move locations, whereas plants have their roots in the ground, sessile, they're not moving.
There's only fight for
They can't
plants.
escape.
There's no fight or flight. Exactly. And it's been that way for ages. And so plants are exceptional in terms of the tools, the capacities they have to fend off threats, because fending off is the only option they have. So for instance, our crop test, we're not going to detect HLB citrus greening. We are not going to be able to, it's not soil borne. However, we can inform what would make for a thriving, healthy plant. Under this framework, we're not just looking at pathogens and biocontrollers, we're also looking at our capacity to cope with stress, our capacity to acquire biologically available nutrients. What are our carbon pathways telling us about the soil's structure and that saturation and drainage issue? So let's take an example. Here we have a corn operation up in the Delmarva area, Delaware, Maryland, Virginia. Yeah, I got that right. And there was one major disease risk here, nigrospora rot. And I'll talk about nigrospora rot. It's a late season pathogen. It comes up in a hot, dry summer condition where the plant senesces. It's no longer defending itself because it senesces. Then you can have nigrospora in hot, dry conditions affect the ears. What about other risks? Okay, we've got low risk of alternaria, low risk of aspergillus, low risk of pythium, low risk of diplodia, low risk of red root rot. We're looking pretty good. Okay, so the only big risk is nigrospora rot. Add in framework number 2. Our capacity to suppress fungal disease is pretty good in the soil, especially in that area that was higher risk. We're looking good so far under framework 2. Is there anything else I can learn from elsewhere in the bee crop test? This happens in hot, dry conditions. Do I have any organisms in here that would support my crop to adapt to droughty conditions? How about ones that improve water use efficiency by producing abscisic acid? Oh, that same area, I have the greatest biological potential to reduce drought stress. That's looking good. Corn gets nigrospora rot when it has senesced early. It's out there in the field awaiting harvest. I haven't got to it yet. Do I have microbes that could actually prolong the life of the plant and delay senescence? Ones that might say reduce ethylene production? Matter of fact, I do. I've got a 57 out of 100 in terms of organisms that reduce ethylene stress. So now I'm feeling super good because my only big risk here is a secondary pathogen, and I've got microbes that can prevent virulence both on the favorable conditions point. I can reduce drought stress, I can slow senescence, and I have fungal suppressive microorganisms. I'm feeling quite good. That's an example of bringing in other elements of the report to inform my crop protection decisions. Same operation, looking at soybean fields. Bacterial wilt, bacterial tan spot are the highest risks on these fields. I've got a sense of where to scout for it. My bactericide agents are low, so this would be vulnerable soil. The only things you could do for a bacterial disease, you can't spray for it. The choices are choose a resistant cultivar, monitor, rotate to a non-host crop. But what if I've already made decisions? I've already planted this crop. I've already got the seed that I've got. Is there anything I can do this season to reduce my risk of the crop suffering from bacterial disease? Okay, let's see. My global stress adaptation potential is quite good. My global plant growth promoting potential is quite good. My plant hormone producers are in good numbers. Global phosphorus, my capacity to mobilize plant available phosphorus is awesome. Pretty good on potassium too. Micronutrients. The best thing I can do to support the immunity of my crop would be to enhance availability of calcium, magnesium, and zinc. Those are the 3 micronutrients that my microbiome is not good at mobilizing on this field. There we go.
Clear instructions.
I've got a decision of how I can protect my crop, not by spraying, but instead by supporting the crop's own immunity through micronutrition. I know we're coming up close on time. I've overshot it, but here's one last case study looking at crown gall in almonds. I've got a lot of crown gall in this one particular field. It was the only field in the operation that had it. What about fungicide agents? Boom. Okay, they're showing up to the fight in a big way. Is there anything else that is tracking this exact same pattern? It's the number of oxygen breathing organisms. There are far fewer oxygen breathers in the epicenter of where crown gall is coming up. Now, crown gall likes aerated soils, but waterlogged or flooded soils and overwatering are strongly linked to infection rates of Agrobacterium tumefaciens crown gall. Saturated soils restrict oxygen. That can weaken plants' roots and cause growth cracks. Those cracks are the perfect entry point for the bacteria. So I have, by branching outside of just the disease and biocontrol metrics, found a plausible hypothesis as to why this one field out of all the others is experiencing higher risk scores for crown gall. And those are the 3 frameworks. Bee crop insights inform more efficient decisions under all 3. And, Sunny and I, all our colleagues, we love to work with clients to understand what are your objectives, and then how can we help you fit what bee crop measures to your operation. For more on this, if you want information about what are they called, pathogen-associated molecular patterns and pattern-triggered immunity, if you want more information about growth defense trade-offs, information about the stress metrics in greater depth, I recommend to you a webinar from last year. You can find on our YouTube channel. What you're not measuring might be costing you yield. We go into much greater depth on this topic in some different facets than we did today. If you want to reach out about bee crop testing, be that bee crop test or bee crop farm, then please get in touch. We'd be happy to support your protection of your crops. And this is where you can find us. And I know I left like 10 seconds in the hour technically, but are we on a hard time limit or can we take some questions?
No, we have a few minutes here. Excellent job, Scott. That was an absolute wealth of information. And thank you to all of our participants and those that submitted questions. We're going to just take one question today and the rest will reach out directly and answer them over email. But I picked the one here that we get the most often and is the most practical of what is the best time of year to test for pathogen pressure in the field?
Sonny, do you want to take it? I've been talking a lot.
No, you go for it, Scott.
Okay. If you are, so I'm assuming this question is coming from a grower. You know, if we were talking to someone who was a crop protection input manufacturer, we would do that in the context of a trial. For a grower, I like to test for pathogens before the season has begun. Sometimes the most logical time to do that is during the fall coming out of the last crop so that we have the winter to make decisions about hybrid variety, resistance traits, make decisions about do we need to purchase seed treatments. You have a whole lot more time to plan when you give yourself a lot of runway. If you have a later planting date, suppose you have a period of weeks after the soil begins to warm back up, you could take a sample closer to planting time. But if you're in a temperate area where you have winter, the soil that you test in fall is going to be a good reflection of the spores and the organisms that will come active in the spring when they're dormant all winter. So my favorite time for most of my clients is fall to make decisions about disease.
Okay, makes a lot of sense. And just to reiterate the difference between our Bee Crop Test and Bee Crop Farm services, the test is just going to be a single snapshot depending on where you pulled the sample in the field. Whereas Bee Crop Farm, we not only give you the spatial analytics and project the results on a map, but we also guide where you should pull the samples based on hundreds of different environmental layers. So it also includes recommendations. So lots of added features with that. We'd be happy to discuss the difference and what's the right fit with you directly.
Thank you, everybody, for your time and attention. And we will reach out about any other questions we got.
All right. Thanks for joining. Have a great day, everyone. Thank you.